Good afternoon. Thank you for joining everyone for our second of the 14-part series. And this is the second one that the three doctors here are going to be providing as the subject matter expertise. We call it the Prayojana series, Liberation of Knowledge, just sharing specifically about COVID and some of the deep knowledge base that they have built together that is going to be useful for anybody and everybody who's out there who's working on COVID or just concerned about COVID. Our subject matter experts are Dr. Joyce Grossman, who has an decades-long experience in U.S. healthcare navigation, disease management. She's also an infectious disease doctor, internal medicine, and pediatrics. Additional research background in immunology, genetics, biochemistry, subbiology. And Dr. Cheryl Ortel, who is a semi-retired OBGYN, who also has a master's in genetics, and who has developed a deep knowledge base in COVID-19 with specifically published evidence-based research. Dr. Adesuwa Okesanya, who is moderating this session. She is a family medicine doctor, an expert in virtual care, and also is a COVID-19 frontline worker who understands what's going on in the everyday lives of dealing with COVID. Thank you, everybody, for doing this. If you don't mind, Dr. Ortel, if you can just go to the next slide.
Hi, everyone. This is Dr. Ortel. We have a little disclaimer. Remember, this is informational only and not medical advice. We're going to be talking about a lot of topics. I wanted everyone to know that there are a lot of COVID-19 resources out there. You can go to the CDC. I especially at WHO like their videos on how to properly take on and take off a mask. The National Governors Association has a lot of interesting information, and clinical trials to see what's out there and can you get a patient on this or what are they trying. Very interesting stuff. So good evening, and we're going to get started. We're going to talk about some medical and genetic perspectives to help all those who are out there practicing. Today's topics are going to include looking at some of the numbers. For COVID-19, we're going to talk about the ABO types, about red blood cells and T cells getting infected, and we'll talk a little bit about the ACE2 binding site and how it allows access into cells, and mention X inactivation. So here from the Johns Hopkins website, we can see that globally we're up to 13 million global deaths, 573,000, and looking like 41 million tests have been done in the United States. So that's a new record for us. We can see that the daily cases in the United States are going up, so that the curve looks like we haven't, for the United States, hit our peak. And we had touched on that last time when we talked about 19 states having some spikes. So here we have cumulative cases by day since the 50th confirmed case. So when it looks like it's zero, there was case. Remember, we also mentioned that the way exponential growth goes, the pattern tends to be case, cluster, boom. And for anybody's area, you could really watch it happen. So that here we have the U.S. in deaths. The curve has started to be not so straight, but cases still going up totally over the United States.
Yeah, I mean, that's been the problem, especially in the South. I mean, the feeling is that they reopened too quickly, and therefore they really didn't, they weren't able, it wasn't contained enough, and then they opened the bars and they opened the indoor dining with air conditioning, and unfortunately, we've seen a resurgence in those areas. which is shown very clearly on the slide.
Yeah, so here we can see Florida get a straight up trajectory, and we can see some of the other states that had been flattened are now going up. This is a reminder, this is the modeled curve of, you know, if we're all the same, we're all little robots, we look the same, we're the same height, we don't have any other morbidities. This is what the stylized version of a pandemic without any social distancing, mask wearing, etc. looks like. And it sort of looks like, boy, Florida's got that angle, doesn't it?
Florida's got it. Florida looks like the prototype of, we're out of control. We don't do social distancing. We don't wear masks. We just hang out on the beach all together, and we just have this straight upward curve.
So in a lot of other places, we looked last time, it looked like New York and New Jersey as a state were starting to really look like this one. Not perfect like this one, but they were coming down. And there's something called Farr's Law, where about 150 years ago, they went by deaths. So they looked at the number of deaths, and it had a nice symmetrical curve. You knew when you peaked, when the deaths started coming down, you knew you were getting, you know, the worst was over. So seeing that a lot of these don't look like they've peaked, we know that there's, in some of these states, there's, the worst is yet to come.
Yeah, I think in Florida now, they're saying initially it was a young person disease, so the death rate wasn't high. But now, because most of these young people are attached to older parents and grandparents, they're now seeing a increase in the number of elderly people, and they're expecting a huge surge in death rates.
Well, actually, that's a great, so that's a great point, because, I think a lot of people have been kind of talking about the death rate, and we're kind of saying, well, yeah, we're getting all these cases, but the death rate is less. And I think some people want to caution on that too, because, it's not really, one, it's not really over. So, and we all know that the cases, testing positive for the cases, the death rate kind of lags a little bit. So that's something also to consider. So while I think I'm all for kind of taking the good news that, oh, well, we're getting, it seems like we have less deaths, I think we also have to be very cautious and careful without kind of making that conclusion, because, it might be a situation where that the deaths are kind of lagging behind.
Yes, and interesting, look at Louisiana. They've got 2 spikes. Now, isn't that interesting? We'll talk about the different clades, further in, but very interesting that this would have such 2 nice spikes there. Mm-hmm. So that could be 2 different strains.
Right.
Yeah,
right.
Definitely. Yeah, that is a very interesting finding. I haven't seen that before.
I've only seen it in England.
Right. And then that was the place that had the 2 strains going on as well. Hmm.
So that's very interesting, because they're the only state that has it. They appear to be the only state that has, it looks like 2 strains.
Well, and we'll have to watch out for California, because we thought they started out with the Wuhan strain, the early strain from China.
Right.
And then Washington was right away looking like Wuhan, and then the European strain is there. So we're not seeing it in new cases. So very interesting, just things to look at. Everybody's got to know the numbers in their own area to see how they're doing, better or worse. All right, I just wanted to remind everybody that the meta-analysis is the best. It's very hard this early in the pandemic to find anything that's a meta-analysis, let alone randomized controlled trials. Some of those are starting to come out. We've seen some in Lancet and that. So here's how the pyramid works. And some of what we're going to present today is going to be expert opinion, background information, and we'll go from there. Just a reminder of the types of evidence. All right. So a nice easy topic to start with is the classical ABO blood grouping system. We know that A and B agglutinogens are complex oligosaccharides differing with their terminal sugars. Antigen A has an N-acetylgalactosamine, and antigen B has galactose. Other than the red blood cell, antigens are also present in salivary glands, pancreas, kidney, liver, lungs, and testes, places like saliva, semen, and amniotic fluid. And that's why you're seeing some of the new tests were coming out, and they were using saliva testing to look for SARS-CoV-2. Now, previous studies have shown, just a little bit of background, that type A, B, and type AB are at higher risk for cardiovascular disease. So just some interest, this is why we're talking about ABO, because there's been other diseases where blood types made a difference. Blood type can influence susceptibility to certain infections, and we see that with norovirus, cholera, and H. pylori, O types are less susceptible, but now in those, they're more susceptible, and only B for the norovirus made you less susceptible. And why is that? So you can see stylized type A will have certain antigens, B will have certain antigens, AB will have them both, O will have none, and then they make antibodies one to the other. So type A can make them against B, B makes them against A, type AB doesn't make any, and type O makes antibodies to both A and B. So in China, so this is a meta-analysis, came out of Iran. So they were looking at all the papers on SARS-CoV and COVID-19 to say whether or not blood groups had anything to do with it. And they right away found that, and it turned out even in the meta-analysis, blood group A is associated with a higher risk of COVID-19 and usually more serious illness. The COVID-19, we know that virus binds to the ACE2 receptor and to ABO carbohydrates. The lack of antigens on the O, so this naked O, makes there less molecular contact with the virus and a lower risk of infection. Blood group O also has lower ACE levels. So ACE is an enzyme that activates angiotensin. Lower levels of this enzyme can reduce the risk of high blood pressure and hypertension, cardiovascular disease. Those are COVID-19 risk factors. The ACE2 receptor also has benefits though. It can attenuate the inflammatory response and redox stress and counterbalances the ACE effect. With lower ACE levels, it works more effectively. The blood group O is also related to higher interleukin-6 levels, and interleukin-6 is a pro-inflammatory cytokine that can be produced by many cells, and it has an important role in the cell defense in the acute phase. So the most important thing that we can say about this slide is blood group O tend to have less risk of infection. That means if you show up at the hospital and you're sick, you're less likely to be type O, you're more likely to be blood group A.
Is it also possible, excuse me, I don't want to interrupt you, but is it also possible, since type O it always has antibodies directed against type A and B, that it's sort of people with type O may be sort of more ready for attack by this virus?
Right, and as we go through this series, we'll always find in this SARS-CoV-2 that there are homologies, there are areas of the gene that can look like certain things. And so, yes, so there's a protective effect against having anti-B because perhaps this bacteria or virus will have something that the B is going to neutralize. So yes, so O would have more antibodies. There's more different antibodies, and there's more ways to get the virus neutralized. So the very first study related to the ABO was the Chinese data, and I went back to that just to show you how absolutely, I thought it was stunning when I read this paper. The Chinese data said that of people showing up who are positive, type A was 63% of them. And so I went back and I looked and compared it to the United States, which has 41% of the population as type A. China only has 28% of their population type A, but in their population, 63% of the positive COVIDs were type A. So something is going on. Type O was seen less often. They looked at 2,200 blood samples. All right, so the Spanish and the Italians looked at it, and I said, well, if we break it out of the meta-analysis, what did that look like? And the blood group O had a 35% lower risk for respiratory failure, and blood group A had a 45% increased risk for respiratory failure. And you know, there's always pockets and cities and that of Spanish, Italian. So I thought it was interesting to look at that when we live in such a diverse country. The lead SNP was identified in chromosome 9 at the ABO blood locus. And that's interesting. And there's a lot of genetic variation there with proagulant markers such as the von Willebrand factor and factor VIII. And so we know that our patients, I'm an OBGYN, we know patients will have SNPs and they'll be more likely to have a bleeding disorder. So these relationships are important to understand. So getting back to the meta-analysis, blood group A is associated with higher risk of infection. COVID-19 binds to both ACE2 and ABO carbohydrates. It lacks lack of antigens on group O leads to less molecular contact. So that's the biggie one. Blood groups O have a lower ACE level. There's the ACE2 receptor benefit and a reminder about the interleukin 6. Since a lot of you may be from the New York, New Jersey area, I included the ABO findings from the from the New York City study with Presbyterian, and they really looked at a lot of stuff that got lost in the meta-analysis. So they associated high blood pressure and O-negative blood type. The oldest patients who were getting infected had these blood types. So the one missing is B. Diabetes mellitus patients were mostly B and A negative blood groups. The people who were overweight and had COVID were mostly the O positive. B was much more likely to test COVID negative, and AB was more likely to test COVID-19 positive. There were no AB negative patients. It's a much rarer blood type, and in the numbers they looked at, there was none. So now we're going to go on. Besides the ABO blood group, there's chromosome 3. There's a gene cluster on there that's associated with SARS-CoV-2 respiratory failure in the Italian-Spanish genome-wide association project that they did. So they looked at 8.5 million SNPs, so single nucleotide polymorphisms found in people who had COVID. So that was 1,610 people in total. And they found that type A was linked to a 50% increase in the likelihood that that patient would need oxygen or go on a ventilator. This did not stay stable when they did the meta-analysis. And the ACE2 genetic variants did not appear to make a difference in the risk of COVID-19. This particular SNP was was associated with the elevated interleukin-6 in childhood obesity of the kids who got sick. So a 50% increase in the Italian Spanish for going to need oxygen or a ventilator. Chromosome 3 is often called the blood type gene. So on that gene is the blood types, but also there's a stretch of DNA that acts like an on-off switch for genes, producing a protein that triggers strong immune responses. So we're going to get more into how being next to something can make a difference and what triggers this on-off gene. And so this is a concern for everyone because that would cause a cytokine storm. If you have an overreaction, you get massive inflammation, you get lung damage, and this is called a cytokine storm, or all these different cytokines, all these different things give you a lot of inflammation. Remember, we talk about most diseases an inflammatory disease. Genetic variation, so SNPs, so those little gene polymorphisms can affect how you respond. There's another locus of 6 genes on chromosome 3 that show an even stronger link to COVID-19, but they haven't told me exactly how that's making your COVID-19 worse so far. So when I was looking, besides the blood type ABO and chromosome 3 perhaps getting triggered, there's something else called glycoprotein OK. Okay, so it's OK is the designation like ABO. It's OK for glycoprotein that's on your CD147s. So what is CD47s? Well, the way SARS can get into, where SARS-CoV-2 can infect your T lymphocytes is going through the novel spike protein on your CD147 membrane. So what happens is you get lymphocytopenia. If you don't have T cells, you can't fight off infection as well. This is a novel invasive route. This was not found in SARS-CoV, but it is found in SARS-CoV-2. They used a pseudovirus to infect permissive cells and also a live virus infection, and they found that the SARS-CoV-2 virus could infect T cells, and much more so than SARS classic, okay? The SARS-CoV-2 infected T cells worked through a receptor-dependent S protein-mediated membrane fusion, and the infection could be inhibited by EK1. So we don't need to get into all that, but I just wanted you to have some background, but that's how that works.
So kind of before we kind of maybe move forward, just kind of like briefly going back on like the blood types, which is kind of fascinating a little bit. I mean, now we kind of dwelt on, okay, A versus O, which seems to be like the two polars. But what about A, B? What about B? Are they kind of like really just sorting in the middle? Like, how are their effectivity?
Right. They're a lot lower. The New York City study really ferreted them out the best of all the other studies. The meta-analysis didn't even touch on this really. So B was more likely to test negative.
Interesting.
Yeah.
That's negative, but they're going to be negative, I'm hoping. This is like, but.
Right. So these are people who infected patients who show up to a tertiary care center, and they say, well, gee, I wonder, what kind of risk factors there are, what's their blood type. So B was more likely to test negative.
Interesting.
I was wondering, the virus does glycolate its spike, proteins. I mean, that's the reason they think that diabetics are increased risk. And I wonder if the glycosylation of the blood group antigens, which is part of the natural being of being a type A, B, or O, has something to do with the infectivity of the virus, since the virus is using the human enzymes to do its own glycylation.
Yeah, it's very fascinating on how this works. And so there's a lot of theories, but trying to demonstrate them experimentally is a lot more difficult.
Oh, I agree, but it's been shown, you know, that glycosylation, at least like for enzymes, is a way of protecting enzymes from being, you know, broken down. So that if I have a protein rather, and it's glycosylated, it has a longer half-life because it's less likely to be attacked by enzymes. So I'm just wondering whether this virus, and of course its nature, has figured out, well, if I use the human enzyme and I glycosylate my spike proteins, I got a much better chance of surviving and getting into that ACE2 receptor.
That's an excellent point. And one of the things I think they're finding is that this particular virus has so many more entry points than any other virus that we really looked at. Very fascinating.
Yeah, and no, it is. I mean, most of the studies look at the spike protein, but that doesn't mean that it doesn't have some other way, some other receptors that it binds with, which are less obvious.
Exactly, and that's why I wanted to discuss some of these points so you could understand how is it getting into the T lymphocytes? Well, gee, we found CD147. A lot of people never even heard of CD147, and that's how it gets in. It also gets in through CD4s, the ACE2. So we keep finding new ways that the way the virus is getting into certain cells. Then, of course, we're going to talk about, you know, kind of globally that people with different genetic variants will be either more susceptible or less susceptible to having different tissue types infected. So we've all seen the ACE2 receptor binding site now probably a lot. So this is the classic. Here's the virus up here, and it's stuck on here. And we're going to get into some changes that affected the way this protein looked. And now the protein used to be very rigid. And when something hit it, it broke right here. But now it doesn't break, it bends. So it'll just flop over and bounce back. But we'll get into that more later. So we wanted to talk about the major route of how SARS-CoV-2 enters cells. And that's by the ACE2 receptor. So first of all, I had mentioned that they were doing a lot more testing using saliva. So let's look at the ACE2 receptors. And if you're going to stick to an ACE2 receptor, where are you going to probably find virus? Well, look where all the ACE2 receptors are. The tongue, floor of the mouth, base of the tongue, and then other sites. So actually this is pretty dense with ACE2 receptors. And that's why people are using the saliva and going to the back of the throat. If we want to look at where in the body are there ACE2 receptors, intestine, kidney, stomach, bile duct, liver, oral cavity, lung, breast, you can read them for yourself, colon, so we see diarrhea, gallbladder, heart muscle. We're hearing a lot more in the news about problems with people's hearts. So colon, gallbladder, heart muscle has a great deal of density. There's a lot of ACE2 receptors. And so in the kidneys, so we hear that there's a lot of problems with kidney failure. So they're side by side with the amount of expression in the organs of ACE2. Breast, ovary, lungs going down, and here's cerebellum. But then we're always going to see people who are having trouble walking, who've gotten infected. So let's talk a little bit more about the ACE2 receptor. It's located, the gene for it is located on the X chromosome. So from data from Italy, we found that there were some rare European variants that were making people very, very ill, and it was explaining the severity in even young people. So this is something, you know, you don't see, most people don't check for it, and young people with these kinds of things would be more affected. So the morbidity and lethality was reportedly higher in men. So they're really going, so what's one, I'll just say what's like one reason why that would be, and that would be because if the ACE2 gene is located on the correct X chromosome, there's going to be less homozygosity in women. So meaning if they've got two X chromosomes and they inactivate one, women are more likely to have activated the one that wasn't useful, okay, or the one that had this problem. X inactivation guarantees a heterogeneous population of ACE2 molecules ranging in production with less, and this was with SARS, binding protein to that allele. So that's all. That's interesting, but that wasn't the whole story. So they looked at genetic variations, SNPs, that increase the expression of the ACE2 in the brain, but not the lung. So here we see all these colors. There's a line here. And what this means is if you're on the left side of the line, you're less likely to bind with the ACE2. And if you're on the right side, you're more likely, and these are SNPs, so variations in the way the ACE2 receptor is expressed. And so they're all yellow. I thought that was nice. So their brain, these are different SNPs. So this is why you're hearing some people will have a lot of mental problems and some really disconcerting things happening to them because their SNPs may be different in different parts of the brain. So here's just a whole list. The only thing that's not brain in here is ovary and anterior tibia, the artery in the tibia. And lung is way over here. So lung is not got that many different SNPs, okay? So basically everybody's got the same SNP, but there's a lot of variation in the brain and how those work. So that was the main take home, that there's a lot of individual variation. And so whether or not this is going to be expressed more or less can affect how the infection will attack you. If it can get into the brain, these are some of the places that people who have SNPs are gonna find the virus attacking.
Well, I mean, this is all very interesting. Of course, you know, we've heard a lot about the ACE inhibitors and if they really have any kind of say with it, especially when the fact that it seems the COVID virus seems to be kind of attacking somewhat through this receptor. I mean, Dr. Grossman, do you have like any thoughts about this as well, about the ACE2 receptor? And
well, I do. I really think it, you know, basically any virus is a relationship between the virus and the host, so that one can't be really looked at without the other. And looking at these SNP variations, it may be possible to predict, depending on the genetic background, whether somebody's going to be more at risk for brain involvement, since everything we get is inherited from our mothers and fathers. And so I would say in a population, if you look and you find, let's say Spain has a SNP that gets a lot of brain involvement, I would expect that that area would show a lot of effects on the brain, seizures, you know, memory loss, and, you know, all the morbidities that you get with this virus. So in a way, that would be,
point.
Yeah. So I think that would help in a more homogeneous population to predict what organs this virus is going to attack. Like in Iceland, which they've been studying and has a very homogeneous population, I think you may be able to predict what parts of the body are going to be, attacked by this virus.
And Joyce, one of the things they've noted that in families, if families have a severe response to the infection, multiple members of the family will react like that. And versus if the entire family has a mild reaction, mostly it'll be mild in the family. And so that could be related. It's related to viral load, but also related to SNPs.
I could also say that in pediatrics, what a finding is, especially with something like chickenpox, which is, of course, a DNA virus, but when one child gets infected with chickenpox, what you'll find is when the other siblings get it, and that's, of course, before the vaccine, the siblings who get it last get it worse. And the feeling was that this virus incorporates into the genome, takes a piece of it. So by the time it gets to the last child, it's very efficient, integrating into the genome and causing damage. So I'm just saying, it could be that in certain populations, this virus is getting better at attaching to certain organs.
Great point, because this is the coronavirus as a group is a very mutating heavy group. So we'll get into a little bit and take a look at the big mutations.
All right. Very quickly, Dr. Ortel, I know someone kind of threw out this question earlier. So the question being that, we've kind of provided very interesting risk information based on the blood types. Now, does it matter about the Rh factor present? If the Rh factor is positive or negative, does that have any role with risk?
New York City's Presbyterian study showed a little bit about Rh factor, but in the meta-analysis, it gets washed out.
I see, interesting.
How about secretors and non-secretors? Because there are, all the blood types have secretors and non-secretors, and I would suspect that may play a role also.
So what she means when she says secretors is those people will be actually secreting the anti-A or the anti-B in their saliva and other different, we had shown a slide with that, but I probably didn't go into that much depth. That those areas will secrete those antibodies, and they can be neutralizing to some extent, which is why you're not seeing infection in the Bs. They have anti-A. Of course, why are we seeing it in the A? They don't have anti-A. They only have anti-B.
Exactly, yes.
Okay, yeah. So I have yet to see any studies on secretors and SARS-CoV-2. But I'm looking,
Would be interesting.
Joyce. Yeah, it would be.
Yeah. Okay, good, because I couldn't find any either. So we're you do a better job than me, so I feel better that there aren't any studies out there.
Oh, I think we have someone who wants to make a comment. I think Dr. Alasala.
Halassa, okay.
Yeah. I don't know if he is. Can we unmute him? Should be you. I think he has a command.
I did.
Can you hear me?
Yes, you can.
Yes, we can.
Right.
Yes.
Yeah, Dr. Ortel, we see the blood type A, it's in the population, the already high percentage than the O. So did the factor of A being the high number among the population is being considered when you say people with A, they tend to be infected more than O? You got my point?
Right, but that slide, it was in the United States, we have 41%. And what was most, I thought, most extreme was China, who had in the 20s percentage of A, and yet 63% of their population that they tested in that study, looking at 2,200 or maybe it was 19,000, I can't remember, but the Chinese, but 63% of the positives were A. So it's going to be several factors. It's not just one. And that, as with anything, it's just one of the factors Blood type A doesn't have anti-A, so they're at a disadvantage. There's other things that put people at a disadvantage, being hypertensive, overweight, having cardiovascular disease, having hypertension. All those things make a difference.
Another thing is that if we say type A, they have more risk of having high blood pressure. So it could be not the reason just because of type A, it would be the factor is hypertension itself. So, you know, there's those factors.
Right, and it is interesting that blood type is associated with cardiovascular risk. Yeah, so there's a lot of different things. So you don't. Well, thank you for bringing that up. Now I'm going to continue with just a kind of an overall review. So COVID-19 predominantly will be a lung infection because the ACE2 receptors are very dense. So there's a lot of ACE2 receptors in the lungs. There's ACE2 receptors in the eyes and the conjunctiva, so you can get an inflammation there. And one of the first reports we had was a Chinese ophthalmologist saying, you know, wear goggles, this thing can infect you through the eyes. We've seen people losing the sense of smell and taste, and I reviewed with you the density of ACE2 receptors in the mouth and the oral cavity. Talked about the lung, the heart. So we're seeing a lot more cases being presented with myocarditis and myocardial infarction. We see that the liver, and half the hospitalized patients are having elevated liver enzymes. One of the factors could be toxic drugs, but still very interesting. And 20% of the symptoms presenting are diarrhea. And we talked that if you don't, if you're not infecting ACE2, how does it get into T cells? Well, it does it by the CD147 antigens, okay? I came across an old study, melatonin indirectly affects ACE2 receptors. So melatonin has a potential antiviral infection, and we've seen it on people's prophylaxis protocols. The United States NIH has a study in the wings using melatonin's sustained release 2 milligrams at bedtime that was first posted in April, but it hasn't opened yet. And then in Italy, and Spain, they were using melatonin as a prophylaxis. So it has anti-inflammatory and antioxidant effects. It regulates the ACE2 expression. It inhibits a couple of proteins, calmodulin, and that acts with the ACE2 by inhibiting shedding of the ectodomain. And that's a key infectious process of SARS-CoV. So this was before SARS-CoV-2, and it reduces replication. So that was one of the reasons people wanted to try it in SARS-CoV-2. So when we look at the difference between SARS-CoV, so that's SARS Classic, that happened in 2002. Now we're talking about SARS-CoV-2, which is started in the winter of 2019. And these are very out of date numbers by now. We went over the numbers of how many there are. They both these viruses could attach to your ACE2 receptor, and that they utilized a serine protease enzyme that can be blocked, and there's a couple of drugs, Camostat, nafamostat, and there's a few more that can block this. And this is an androgen-dependent enzyme. So one of the reasons when we were hearing in Italy and Spain that there were so many males was this is androgen-driven. So if the more testosterone, the more androgens you have, the more this was expressed, and it would go snip the spike protein, so it would be better able to attach. And they used people's SARS convalescent sera, and they tried it, and it works really well on SARS-CoV, but of course SARS, right, from 2002, that sera, if you'd had it, doesn't really protect much against your SARS-CoV-2. So here's a nice look at genetic variants that are associated with higher ratios of ACE2 cells. So the more ACE2 receptors you have, the more sites you have that the virus can bind to. And at the top of the list, Japanese and Han Chinese. So very interesting. And then down at the bottom of the list, Tuscany in Italy, those people had 51% density compared to 92%, so quite a bit less ACE2 receptors. And then you can look for yourself for a minute, all the different variations there are in between, and this is why, you know, you ask, what's your ethnic background and how likely are you to have more ACE2 receptor densities? These are all the different SNPs that they looked at. And I'm thinking probably if you've had your 23andMe and you looked at the raw data, you might be able to see whether you had these SNPs. Very interesting. Now we're going to talk about the furin cleavage site causing an increase in neural symptoms. So this is real interesting. The furin cleavage site is something new, so it hadn't been in SARS, and we find it just popping up in SARS-CoV-2. It helps the virus to fuse to the host cell and membranes, so it's easier for the virus to get in. And it doesn't need to get in, so this is before you've infected the cell. It uses inside the body, it uses the host enzyme furin to do the cutting for it. So it doesn't have to use any of its own energy. It doesn't have to be transcribed or anything. It uses the host furin to snip it, and so it can get in, then it can get in and merge with the cell and infect it. So they're looking at some furin inhibitors. And one of the interesting things, and a study came out in the fall, I think it was October 2019, the Chinese had put a furin cleavage-like site into an infectious bronchitis virus for chickens. And then they noticed that this, by doing this, they had pronounced neurotropism in the chickens. And I've seen infections like this in mice when I was an undergrad. Like they walk around in a circle and they'll go like that with their head, they'll tip it. So I just wanted you to remember the slide that I gave you to prepare you for this was that there's all kinds of SNPs in people. So genetic variations can change how much the ACE2 is expressed in the brain. So we looked at the brain was one of those tissues that had an awful lot of polymorphisms, and that's why this can be so dangerous. We're going to get back to the TMPRSS2, and that's the androgen-driven serine enzyme. So it's on chromosome 21. The androgen receptors on chromosome X, and the ACE2 receptor gene is on chromosome X. So you get activated. It's primed by the androgen. It snips one of these spikes, and it makes it so it's better able to attach. In Italy, they had a nice paper where they looked, because it was affecting more males. So everybody's going, how come, how come? And sex differences in ACE2 levels didn't explain the differences in the disease, so they was trying to see what was it. And they looked at the different SNPs, and they decided that it was really the SNPs and the TMMPRSS2 expression. And there was one SNP that was a lot, was the worst. And higher levels of this particular enzyme are more frequent in Italians. And of interest to people who have a lot of different types of patients, in the East Asian population, there's no expression of it. So that was fascinating. Older postmenopausal women lack estrogen, so they lack the protective effect from this estrogen, and they also tend to have lower androgen levels. So that's another reason you'll see sex differences. Any questions? Okay.
Oh, so I actually wanted to kind of comment on a little bit on that. I mean, it's sex differences. So generally, am I right? I thought we saw more, I feel we saw this virus seems to have affected more men than women. Is that really true? Though that is what I've heard.
Right. So that's true, and they're wondering like what it would be. And so I'm trying to dig down, and they said, well, it's not the ACE2 receptors because there's no sex difference between
Right.
the density of ACE2 receptors, but they did find this one particular enzyme. It's a transmembrane protease that SNPs the spike protein, and it's stimulated and made more. Okay, so there's more of this enzyme around if you have more androgen around.
Ah. Okay.
So that's one of the reasons that will cause men to have more of a viral load for the same amount, because now you've got an enzyme that's going to be more, that's helping the virus get in.
Yeah, that makes a lot of sense, yeah.
Yeah, so this enzyme's in the prostate, colon, stomach, and salivary gland.
Mm-hmm.
So the androgen effect would be seen there. I thought that was interesting when I read it.
Absolutely. I think we have another question actually from our Dr. Halasa. Is he ready to speak?
Yeah.
Yeah, Dr. Ortel. The other thing is we find out that male, they have high risk of having cardiovascular, especially comparing to the female, because they are protected, the females, by the estrogen. So the estrogen is playing a role in protecting women from COVID-19 for many reasons. Even they don't get complicated because of estrogen do inhibit interleukin-6. So I think estrogen does have protective effect against COVID-19. Estrogen does have protective effect against cardiovascular diseases. Cardiovascular disease has high risk of you get infected and complicated, because we know those COVID virus do affect the endothelium of the blood vessels. That's one of the targets. And most of the complications, possibly it's due to the damage of the endothelium of the blood vessels. And patients with hypertension and diabetes and obesity, they already have their endothelial dysfunction. So if you see those and you're trying to connect, I think it's better to end up into the common things, so that those multiple factors will not confuse the whole situation. You know, in other words, if we can link it to finally to the endothelial dysfunction, then that makes sense why the male versus female, they have higher risk of COVID-19. And if we link the estrogen with cardiovascular, with endothelial function, So we can narrow it down to really determine exactly what's the main factor in COVID-19. I think endothelial dysfunction is the main factor, whether it is risking of getting affected or complication. Do you got my point?
Yes, I get your point. I'm just setting the stage. I think it'll be excellent to have a topic on endothelial dysfunction in the future. I'm just trying to get everybody to get some background. Thank you for your point. Very good. I want to talk a little bit about the mutations. We've already mentioned that coronaviruses mutate a lot, and there have been over 10,000 mutations in this 30,000 amino acid virus. And I want you to understand that when we talk about these things, what a little piece we're talking about. So we're going to talk about the G clade spreading, and it's now 70% of all the cases are G clade, but this is based on testing 0.6% of the global cases. All right, and who does all this testing? So there's a global initiative that shares all influenza data, and it's out of Germany, Singapore, and the United States, and supported by private and corporate funds. And so we talk about the Wuhan strain to make it easy, the one that came first from China. As soon as it got out of China, pretty much, it started mutating, and it mutated, went back in. And so now we're just going to kind of see how this thing has been traveling. So what's a G clade? That means there was one amino acid substitution at number 614. So if you numbered all the 30,000 base pairs in the SARS-CoV-2 virus, there, instead of a D, there was a G, okay? So then very early on, we said, well, does this make any difference? And this is the one I said, yes, it looks like it does make a difference to have this thing change from a D to a G. And so if we look at globally, here we started out with the Wuhan, original Wuhan, all right? And now this, so that was where there was a D. And now we can see where there's a G is blue, and that's most, it's taken over, and that's about 70% of the cases. So in Europe, it looks like this. And in North America, we started out on the West Coast with the Wuhan, the D strain, and now we have what we tend to say is the European strain or the G clade. Very interesting in Italy having two, this looked like Louisiana. So one wondered if there was something else going on. And here we have US, New York, Washington. Look at New York. All right, so there's one paper from Northwestern. So Northwestern University is in Chicago. So they were looking, and they were doing this kind of research where they're looking at the cases, and they looked at 88 people who came in, and they said, we'll do the genotype of the virus and see where it is coming from. So clade 1 looked like New York City. Clade 3 was coming from Washington. And clade 2 was the China, Chicago thing. All right, so here in 88 patients from Northwestern, so 88 patients from that Chicago area went and got tested. They had SARS-CoV-2 and they checked the genome of it. This was from Washington State. This was from New York City. And then this was a new clade. Scripps came out in June, and everybody's asking, does this D to G change make any difference? Is it increasing infectivity? Well, they did this in a Petri dish. So this is a Petri dish. They looked at it, and they said that it's that because of the furin cleavage site stability. So this is like, instead of breaking when something hits it, it bends and it pops back because of the salt bridge. It's 9 times more infective. Okay, that's in the Petri dish. So why is, the later virus work? Could be this. I'll give you other ideas on this, but okay. But one, they proved in a Petri dish that yes, having this change makes it more infected. Okay.
Dr. Ortel, it's Juby. I'm so sorry to interrupt. We are at 8 o'clock, so I just wanted to let you know we're going to extend it to 10 more minutes if everybody's okay with it. Are you guys all okay on the time in terms of, and if you're not, please drop, whoever is attending, if they don't have the time. We will be also sharing a video version of this on YouTube. So I'm sorry to interrupt, but continue on.
That's good. There's only a few more slides. So additional stability was needed. That's what is supplied with this D to G change. It has to do with positive and negatives. But there's another thing that's going on in this G clade, and that's that there's a new stop codon. So there's 3 amino acids got put in there, and it tells the when it's being transcribed, it says to stop. And so what happens is it makes a long protein and a short protein. And the long and the short of it is, we don't know what that short protein does. And could that be the reason that this is more infective? So that's another reason behind the G clade. And in the future, we may see that this G becomes G little r or G little h. And some of these, like the hitchhike protein, the hitchhiker gets hooked on there, and they say, no, it's actually, there's 3 or 4 mutations that are making this a lot worse than what we thought. And just a reminder that genes next to each other make a difference. So they can make things better, they can make things worse. And we saw that besides this spike D614G, there is another S9291 in Sweden, Wales, and England that looks like it was increasing infectivity. So this G clade may have more mutations in it that is making things worse. So in conclusion, we covered what was new in the cases. We looked at ABO types. We looked at how the virus can infect and attach to RBCs and T cells. And we looked a little bit more at the ACE2 binding site and the new G clade, and there's probably more mutations, and are they going to be of benefit or make things worse? Well, of course, in medicine, we're going to be studying the things that make it worse, right? If it made it better, it made it less virulent, you're not going to show up at the hospital.
Exactly.
Please stay safe and know your risk. Any other questions?
Yes, there was a question about, I'm just going to read it, Adesuwa, just because I can see, from Mandy asking if you can comment about the prospect of developing any meaningful immunity passports.
Well, that's a can of worms, and we will go into different things in future sessions, but the problem with SARS-CoV-2 is that it's mutating so quickly. So will the vaccines work against the new mutations? That's one. Number 2, it looks, all coronaviruses, our immunity to a cold doesn't last that long, 3 to 6 months. So if immunity to one of the strains lasts 3 months, and you had the Wuhan original strain, and then you got the G clade strain, will you be immune against it? We don't know. Some early data out of China said 7% were not immune. 7% who got reinfected were not immune. And so we're thinking that maybe, they had the European strain hit them after they had the China strain. So they had the 614, and then they got the G614.
There's some studies going on in Spain and Sweden, and they're looking at T cells, and they feel that T cells play a very important part in immunity, and that more people may be immune than we first thought because people had T cell immunity from other coronavirus infections.
Excellent point. So what they're saying is even if you don't have antibodies, you may have memory cells that will be activated when you're reinfected.
Right.
However, you know, that can be a two-edged sword because sometimes when SARS, we tried to make a vaccine, and in MERS, when we tried to make a vaccine, reinfection triggered cytokine storms. So there's a lot we don't know yet, and it's hard to measure, you know, a memory cell until you've actually infected somebody, and so that's really an unethical study.
Yeah, well, dengue fever is the same thing. It's the reinfections which lead to terrible disease, and that was the problem with the vaccine.
Yes, that vaccine went to the Philippines, and if you were vaccinated and then got dengue fever, you were one sick person.
Really much sicker. Yeah,
I mean, they had 600 deaths.
Yeah, no, it was a real surprise because I remember the dengue, it worked so well. And then in, you know, real time, you know, when you got infected again, it was a cytokine storm that killed you, which is the problem with native dengue fever. Dengue fever, when you have it for the first time, is breakbone fever. You know, like COVID-19, you wish you were dead, but you usually survive. It's when you get reinfected that you don't survive. And unfortunately, the vaccine, you know, did the same thing.
Yep, got lots of interesting topics that everybody's going to be covering in the future here. So stay tuned. Remember, we're trying to give you some information. We're not trying to give you medical advice. If you have any questions or comments, please send them to inquiry@globexhealth.com, and we'll be happy to answer them either individually or at our next session. Thank you so much for attending, and thank you to all the people who had comments. Again.
Thank you, Dr. Grossman, Dr. Ortel, and Dr. Okesanya for doing this. So this was the second part of the three-part series with the three of these experts. Then after that, we're going to dive into several different tangents. You have heard Dr. Halasa here. He will be presenting in one of the sessions in the future. He's a wealth of knowledge. So we will be posting this on in YouTube under, I believe, Globex Health or somewhere. So, and the slide deck from last session and this session will be shared with you in the next 2 days. We need to kind of make sure that we edit out or we only include what we shared. I believe this particular session we are able to do that. If there are any other questions, let me see. Was there any other question, Christina? You're mentioning there is a question here that I didn't bring up. Was there, Debbie, did you want to ask a question? Because I don't know, I didn't see it. Let me just see. Oh, I see that. I unmuted you. You feel free to ask the question.
Oh yeah, okay. Hi, Dr. Grossman. I'm glad to hear your wonderful voice.
Thank you.
I used to work for you in Blue Cross. Hi, Dr. Ortel and Dr. Okesanya. What a wonderful topic you guys are discussing right now. So I'm working right now in COVID-19 hotline. So it's a lot of eye-opener. And of course, I'm working not in the East Coast, in the Midwest setting. I've been seeing work exposure, community exposure is definitely, there's a surge here with us as well. But with the household members, I've been seeing somebody, let's say a mother, who got infected and did not get better and actually got worse Then few days after, or probably a week after, a husband or a son also got the virus. So how would you know if this is a continuation of her disease primarily, or this is reinfection?
Oh,
Well,
that's Dr. Ortel, do you want to take that? Because that is a raging debate now.
Mm-hmm.
I'll try it because I'm an immunologist, but I did some work in it. Again, at this point now, no one is actually labeling it as, a reinfection or a new infection. We really don't know enough. Remember, this is a new virus, and it's cropping up in new ways. So it's difficult to say. I could say that if it's a reinfection, it means that we have a real problem with immunity. And unfortunately, the studies from the UK back that up, because if the antibodies are giving protection for only 2 or 3 months, I would say they're beginning to wane, and the person now is able to be reinfected if the T-cell immunity is not strong enough, isn't giving enough of a memory. And that may be genetic.
Sure.
It can be genetic, it can be load related. So if you had a small load, your humoral immunity would take care of it, and you wouldn't have time to make antibodies.
Sure. Yeah, wow. It's interesting. Thank you. It's very helpful.
We love to be of help, at least.
Since if anybody else have any questions, let us know. I can open up I open it up for it. Otherwise, I just wanted to thank the doctors for sharing their knowledge base. Thank everybody for attending and just allowing us to, kind of bring in what we call knowledge liberation. We just want you to share this knowledge and utilize it so that we can bring these numbers down and potentially mitigate, the second wave potential that may happen in the fall. So thank you, everybody. Look forward to the next session and have a good evening.
Good night, everyone.
Night.